This is a biology praxis exam prep unit on evolution. First, we're going to look at a summary of evolution, and then we'll do a series of practice questions that are similar to what you might see on your biology praxis exam. Evolution summary. Distribution and movement of alleles. Genetic variation describes naturally occurring genetic differences among individuals of the same species. Allele frequencies in populations are caused by mutations, which is a spontaneous change in DNA, by gene flow, which is movement of individuals into or out of a population via maybe migration, for example, genetic drift, which is a change in allele frequency due to chance, non-random mating, where individuals are choosing mates based on specific traits leading to sexual selection. Sexual selection is a mode of natural selection. Members of one biological sex choose mates of the other sex to mate with and compete with members of the same sex for access to members of the opposite sex. Genetic drift. Genetic drift happens when the allelic frequencies in a population change due to chance. The smaller the population is, the greater the impact of genetic drift. With a small population, it might only take a few generations before they are all the same color. As you see down here, we start with an even split in the original population between red and blue, and over the generations you can see the drift more and more towards blue color. Genetic drift tends to eliminate variation more quickly in small populations. Large populations tend to have greater genetic diversity. So in this small population, it takes 5 generations to get to a population that is entirely blue. Bottleneck effect and founder effect. 2 types of genetic drift are: 1, the bottleneck effect, where most of the population is killed off due to a chance event such as fire, flood, volcano, or earthquake. As a result, all of the alleles carried by these individuals are lost. Because there are so few individuals left, there's decreased genetic variability. And 2, the founder effect, where a very small group of individuals populate a new area, forming a new group, and they are the founders of the new population. Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium measures microevolution rather than large-scale macroevolution. Microevolution is the change within small groups of organisms or species over a short period of time. Macroevolution is the evolution of large groups of organisms over a long period of time. By measuring allele frequencies, how often dominant and recessive alleles show up in populations from generation to generation, the Hardy-Weinberg equation can be used to determine if a population is currently evolving. Conditions for Hardy-Weinberg. The Hardy-Weinberg equilibrium equation can only be used with the following conditions to determine if the population is evolving. Populations have to be extremely large. Allele populations are stable. There are no mutations. Mating is random, and there is no natural selection. Hardy-Weinberg equilibrium is calculated using the following equations: p squared plus 2pq plus q squared equals 1, and p plus q equals 1, where p squared is the frequency of the homozygous dominant genotype, and p is the frequency of the dominant allele. 2pq is the frequency of the heterozygous genotype, and q squared is the frequency of the homozygous recessive genotype, while q is the frequency of the recessive allele. All right, let's look at an example using the Hardy-Weinberg equation. A population has 36% white flowers, which is homozygous recessive? What percent of the population will be homozygous dominant and heterozygous? So on this slide, we're going to look at homozygous dominant. We'll answer the first part of the question. And on the next slide, we'll answer the next part of the question. All right, so what are we given? This 30% white flowers, which we're shown is homozygous recessive. That is the frequency of the homozygous recessive genotype, Q squared. All right, so once we know Q squared, we can do some calculation, but also what are we asked to find? The percentage of the population that's homozygous dominant, which is Q squared. So we're given P squared, we're given Q squared, and we're looking for P squared. So we can take the square root of Q squared to get Q equal to 0.6. That's the frequency of the recessive allele. Okay? Then we can use that second equation and find that P is equal to 1 minus Q, or 1 minus 0.6. That gives us 0.4. That's the frequency of the dominant allele. And then if we square that, we get 0.16, which is the frequency of the homozygous genotype, capital B, capital P in this case, or P squared. Okay, so we're going to take all four of these values, P squared, Q squared, P, and Q, and take them into the next slide to answer the second question: what percentage of the population will be heterozygous? All right, so now we're going to calculate the frequency of the heterozygous genotype, where we have capital P, lowercase p. So we have the Hardy-Weinberg equation, P squared plus 2PQ plus Q squared equals 1. And it's important to note that this right here is the frequency of the heterozygous genotype. So that's what we're trying to solve for. So I just moved the other terms to the other side of the equal sign to find that that frequency is 1 minus P squared minus Q squared. Now Q squared was given in the problem, and Q squared we found on the previous slide to be 0.16. So 2PQ is equal to 48%, 0.48. And that's the frequency of the heterozygous genotype. So the percentage of the population is 48% heterozygous, 16% homozygous dominant, and 36% homozygous recessive. So lowercase p, capital P, and P. Evolution through natural selection. Natural selection is when organisms that adapt better to changing conditions survive, reproduce, and have more offspring. Natural selection requires variations in traits, inheritance of traits, competition for limited resources, reproduction success differences, and deep time. Artificial selection. Artificial selection is another type of evolution, but is driven by human input. Humans control breeding to select favorable traits in offspring. Starting 7,000 years ago, humans have bred dogs by choosing which pairs will mate and pass on their desired traits to the next generation. Over many generations, the diversity in the dog population has increased markedly. In the case of the image shown, the ancient ancestors of this Jack Russell Terrier were wolves. Evidence. Populations change over time. Evidence that supports the claim that populations change over time are things like fossils, anatomy comparisons, DNA and protein sequence comparisons, animal embryo comparisons, and biogeography. Fossils. Fossils are preserved when sedimentary rock forms around the remains of living things. Very old layers of sedimentary rock can be lifted to the surface by plate tectonics, giving us access to old fossils. We know how old fossils are because we can tell the age of the rock in which they are preserved. Anatomy comparisons. Anatomy is the study of body structures. When we compare the anatomies of different types of organisms, we see similar structures used over and over again, but in each species, the structure may have changed over time and become adapted to carry out different functions. The best explanation is that they evolved from a common ancestor with these characteristics. This is evidence for descent with modification. Homologous structures are parts of animals that look similar to parts of other animals. Even if they perform different functions, they are evidence for a common ancestor. Analogous structures are parts of animals that do not look similar to parts of other animals, but are similar in function. They do not have a common ancestor. Vestigial structures. Vestigial structures are structures on an organism which have lost function and atrophied or shrunk through time. The body part had a function in an ancestor, but through modifications and evolution, the body part is no longer useful. So you can see in this flightless cormorant, the wings have shrunk down because they don't have the same use or they don't have the same need for them as an ancestor did. DNA and protein sequence comparisons. All species share genes required to carry out basic cellular processes. By comparing these gene and protein sequences, we can gather evidence about how closely related one species is to others. Like anatomy comparisons, DNA and protein comparisons give evidence for descent with modification. Animal embryo comparisons. Very early in development, when an animal is an embryo inside an egg or a uterus, the embryos of different species resemble one another. All animals share similar genetic programs that control early development, which is strong evidence that all animals share a common ancestor. As we grow and develop, other genes go to work and species' unique adaptations become more apparent. Biogeography. Biogeography is the geographical distribution of species. Darwin noted that the species on the Galapagos resembled the species on mainland South America more than species on other groups of islands. Because of this evidence, Darwin proposed that the Galapagos species must have come from a common ancestor of the species on the mainland of South America, or that the Galapagos varieties evolved from the mainland immigrants. Modes of natural selection. There are 3 modes of natural selection. Stabilizing selection favors intermediate variations and reduces variation in a population. Disruptive selection favors one of the extreme variants in a population and occurs when there is an environmental change or when individuals of a population migrate to a different environment. And directional selection, which favors the extreme and leads to increased variation. Speciation occurs when this variation is so great that a new species is created. Types of speciation. In allopatric speciation, groups from an ancestral population evolve into separate species due to a period of geographical separation. In sympatric speciation, groups from the same ancestral population evolve into separate species without any geographical separation. Causes of speciation. Prezygotic isolation occurs before the formation of a zygote can take place and include behavioral isolation, where 2 populations are capable of interbreeding but have behaviors that prevent mating. Geographic isolation, where 2 populations are separated by geographic barriers like mountains or bodies of water, temporal isolation, where two different populations mate at different times, or mechanical isolation, the sexual organs between members of closely related yet different species do not fit together. Post-zygotic isolation occurs after members of two different species have mated and produced a zygote and include infertile offspring or offspring dying before sexual maturity. Modes of evolutionary rates. Most evolution is gradual, small changes over long periods of time, known as gradualism. But in some cases, new species can appear or disappear suddenly. This type of evolution is known as punctuated equilibrium. The sudden appearance or disappearance may not be as sudden as thought. Speciation may have occurred during time periods too short to be distinguished in the fossil layers. Patterns of macroevolution. There are 4 types of macroevolution: adaptive radiation, divergent evolution, convergent evolution, and coevolution. Patterns of macroevolution. Adaptive radiation is the emergence of numerous species from a common ancestor. This generally occurs when organisms move to a distinctly new environment from the old one, or when there is major change in the current environment, as in Darwin's finches. Adaptive radiation leads to divergent evolution, new species arising from another species. Divergent evolution can arise from disruptive selection. Convergent evolution is when different organisms, arising from different ancestors, have similar characteristics because they adapted to similar environments. For example, dolphins and sharks. Coevolution is when an evolutionary adaptation in one species affects the evolution of another species. For example, pollinators and flowers. Major evolutionary trends. Major evolutionary trends support evolution. The first animal trait to evolve was multicellularity. This was highly adaptive. Multiple cells could do different jobs. They could evolve special adaptations that allowed them to do their job really well. Another trend is cephalization. This is the concentration of nerve tissue at one end of the body, forming a head region. This is highly adaptive. It allows central control of the entire organism. Cephalization was the first step in the evolution of a brain. The theory of endosymbiosis. The mitochondrion was once a bacterium that efficiently converted glucose to a simpler form of energy. The chloroplast was once a bacterium that could perform photosynthesis and create glucose. The theory of endosymbiosis is consistent with the process of natural selection. In each case, the prokaryotes were more successful together than they were apart. At first, they formed mutualistic symbiotic relationships. Both chloroplasts and mitochondria have the 4 structures that all prokaryotic cells have: their own membrane, cytoplasm, a circular chromosome, and ribosomes. They also reproduce within eukaryotic cells. So the evidence for the endosymbiosis is the containing of these 4 prokaryotic cell structures. And it kind of looks like this, where we have a host cell that engulfs the bacterium. So now it has a second plasma membrane, the vesicle membrane, and then the original bacterial plasma membrane. And then over time, we get the evolution of the inner and outer mitochondrial membranes. Origin of life on Earth. Theory 1, organic monomers from space. The dust in the solar system from which Earth formed was rich in organic chemicals. Meteorites striking Earth would have hit with lower velocity since the atmosphere was thicker. The monomers would have survived. This is also known as panspermia. Theory 2, organic monomers from reactions in the shallow ocean. In 1953, Stanley Miller tested a hypothesis involving an artificial mixture of inorganic molecules while simulating the conditions thought to be found on primitive Earth. Within days, the experiment produced some of the 20 organic monomers presently found in organisms, as well as other organic molecules. RNA. The chemical reactions in cells would eventually create sugars and then ribonucleic acid, RNA. RNA is capable of enabling key functions of life: replication, making identical copies of itself, energy storage for chemical reactions, and catalysis, dramatically speeding up favored chemical reactions. Once these three functions were developed, evolution accelerated. In more advanced biological systems, RNA's functions have been taken over by more specific chemical processes. Atmospheric conditions. Photosynthesis takes sunlight, carbon dioxide, and water, and produces sugar and oxygen. The evolution of photosynthesis contributed oxygen to Earth's early atmosphere and helped change it from one rich in carbon dioxide, like you see on the left, to one rich in oxygen, like you see on the right. Factors that led to extinction. The founder effect and the bottleneck effect lead to reduced genetic variation within a population that can make a species less able to adapt to changing environments and therefore less likely to survive and reproduce. Scientists estimate over 99% of all species that have ever existed are now extinct. Extinction can also be caused by a variety of other factors, including meteor impact, human impact, climate change, predation, disease, interspecific competition, and degradation of habitat. The sixth mass extinction. Many scientists have proposed that we are in the midst of the sixth mass extinction, called the Holocene epoch, which started 11,700 years ago and is related to the impact of human activity and population growth. The extinction rate is about 1,000 times what it would be without human impact. The extinction of many species of animal and plant life is attributed to human hunting and climate change.